Hi friends, today we are trying to solve some questions on biotechnology and its applications. If you want to try this yourself, then mute the video and then solve this one by one. So, let's start. First question. Study the following statements regarding Bt-toxins produced by bacterium Bacillus thuringiensis and select the correct one. Option A. Most strains of Bacillus thuringiensis produce proteins that kill certain insects such as lepidopterans, coleopterans, and dipterans. Option B. Bt-toxin protein do not kill the bacteria themselves because the toxin proteins occur in an inactive form called protoxins. C. When an insect ingests the inactive Bt-toxin, it is converted to an active form of toxin due to alkaline pH of the gut, which solubilizes the protein toxin crystals. And D. All of the above. So what will be the answer? The answer will be option D. Soil bacterium Bacillus thuringiensis produces proteins that kill certain insects like lepidopterans, coleopterans, and dipterans. Lepidopterans means tobacco budworm, armyworm. Coleopteran means beetles, and dipterans means flies, mosquitoes. Bacillus thuringiensis forms some intracellular protein crystals. These crystals contain a toxic insecticidal protein. The Bt-toxin protein exists as inactive toxin and is converted into an active form due to the alkaline pH of the alimentary canal that solubilizes the crystal. The activated toxin binds to the surface of the midgut epithelium cells and create pores which cause cell swelling and lysis and finally cause death of the insect. Therefore, the option will be D. Next question, question number 2. Bt-toxin gene has been cloned from the bacteria and expressed in plants to provide resistance to insects without the need for insecticides. Examples of such plants are option A. Cotton and corn, B. Rice and potato, C. Tomato and soybean, D. All of these. The answer will be option D. All of these. Next. Read the given statements and select the correct option. Statement 1. Ti plasmid obtained from Agrobacterium tumefaciens is effectively used as vector for gene transfer in plant cells. Statement 2. The part of Ti plasmid transferred into the DNA of plant cells is called as tDNA. So which will be the correct answer? Option A. Both statements 1 and 2 are correct and statement 2 is the correct explanation of statement 1. Option B. Both statements 1 and 2 are correct, but statement 2 is not the correct explanation of statement 1. C. Statement 1 is correct and statement 2 is incorrect. D. Both statement 1 and 2 are incorrect. So here the answer will be option B. Both statement 1 and 2 are correct, but statement 2 is not the correct explanation of statement 1. Ti plasmid, tumor-inducing form, the soil bacterium Agrobacterium tumefaciens, is effectively used as vector for gene transfer to plant cells. This bacterium induces tumors in broadleaf plants such as tomato, tobacco, and soybean. The plasmid is disarmed by deletion of the tumor-inducing genes. This transformed bacteria can still infect plant cells. The part of Ti plasmid transferred into plant cell DNA is called tDNA. This tDNA with desired DNA spliced into it is inserted into chromosomes of host plant where it produces copies of itself by migrating from one chromosomal position to another at random. But it is no longer produces tumor. Next question 4. Though Green Revolution has been a resounding success in terms of agricultural production, yet it has failed in its overall social objectives because option A. It has not succeeded in making India totally and permanently self-sufficient in food. B. Use of agrochemicals becomes very expensive for Indian farmers as well as these have harmful effects on environment. C. In regional terms, only Punjab and Haryana states and eastern plains of river Ganges in West Bengal state showed reasonably good results, but results were less impressive in other parts of India, and option D, all of these. The answer will be option D, all of these. Next, which of the following statement are correct with regard to the disadvantage of GM crops? A. GM crops can affect human health by causing allergic reactions. B. Transgenes in commercial crops can endanger native species. Example, the Bt-toxin gene expressed in pollen might endanger pollinators like honeybees. C. Production of GM crops cause damage to the natural environment and is always costly. And D. All of these. The answer will be D. All of these. Next, study the following statements and select the incorrect ones. 1. Bt in Bt cotton indicates that it is genetically modified crop produced through biotechnology. 2. The anticoagulant hirudin is being produced from transgenic brassica and apple seed. 3. Flavor-saver transgenic tomatoes remain fresh for a longer period than the normal tomato variety. And 4. Golden rice is a transgenic variety of Oryza sativa which is rich in beta-carotene and helps to prevent night blindness. So what will be the answer? The answer will be option A, one only. Bt in Bt cotton stands for Bacillus thuringiensis. Next, which of the following risks are associated with genetically modified foods? A. Toxicity in human beings. B. Allergic reactions in human beings. C. Antibiotic resistance in microorganisms present in alimentary canal. And D. All of the above. So the answer will be option D, all of the above. Next, flavor-saver variety of tomato which remains fresh for a longer period than normal tomato variety. Option A has high amount of enzyme polygalacturonase. B has reduced amount of enzyme polygalacturonase. C is a pest-resistant variety. D is rich in vitamin A and prevents night blindness. So the answer will be option B. The tomato variety flavor saver presents an example where expression of a native tomato gene has been blocked. Fruit softening is promoted by the enzyme polygalacturonase, which degrades pectin. Production of polygalacturonase is blocked in transgenic tomato variety flavor saver. So, fruits of this tomato variety remain fresh and retain their flavor much longer than do the fruits of normal tomato varieties. Additionally, the fruits have a superior taste and increased total soluble solids. Next, which of the following statements are correct regarding the process of RNA interference? 1. This is used to prevent the infestation of protozoans. 2. It takes place in some eukaryotic and all prokaryotic organisms as method of cellular defense. 3. The method involves silencing of a specific mRNA due to a complementary double-stranded RNA molecule. 4. It is a novel strategy to produce pest-resistant plants. So what will be the answer? The answer will be option A, both 3 and 4. RNAi or RNA interference takes place in all eukaryotic organisms as a method of cellular defense. This method involves silencing of a specific mRNA. Using agrobacterium vectors, nematode-specific genes are introduced into the host plant tobacco plant. The introduction of DNA was such that it produced both sense and antisense RNA in the host cells. These two RNAs, being complementary to each other, formed a double-stranded RNA that initiated RNAi. Next, genetic engineering is possible because option A, we can cut DNA at specific sites by restriction endonuclease. B, restriction endonuclease purified from virus can be used in bacteria. C, the phenomenon of transduction in bacteria is well understood. D, we can see DNA by electron microscope. So here the answer will be option A. Next, which of the following genes were introduced in cotton to protect it from cotton bollworms? Option A, cry-AC and cry-AB. B, BTAC and BTAB. C, cry-1AC and cry-2AB. And D, NIF genes. So here the answer will be option C. Two cry genes, cry-1AC and cry-2AB, have been incorporated in cotton. The genetically modified crop is called Bt cotton as it contains Bt-toxin genes. The genes cry-1AC and cry-2B control cotton bollworms. Next, Bt-toxin proteins crystal present in bacterium Bacillus thuringiensis do not kill the bacterium themselves because A. Bacteria are resistant to the toxin. B. Toxin occur as inactive protoxin in bacteria. C. Bacteria enclose toxin in a special sac. D. None of these. So answer will be option B. Next, golden rice developed through transgene approach is enriched with A. High lysine content, B. High methionine content, C. High glutenine content, and D. High vitamin A content. Here the answer will be option D. Rice is a staple food in many countries, particularly in Asia, but doesn't contain vitamin A or its intermediate precursors. By inserting two genes from daffodil and one gene from a bacterial species into rice plants, Swiss researchers have produced rice capable of synthesizing beta-carotene, the precursor of vitamin A. This rice is called golden rice because of yellow color of rice grains due to the presence of beta-carotene. Next, all are the biotechnological applications in order to increase food production except A. Apiculture, B. Agrochemical based agriculture, C. Organic farming, D. Genetically engineered crop based agriculture. Here the answer will be option A. Next, agrochemical based agriculture includes fertilizers and pesticides, genetically modified crops, RNA interference, and all of this. Here the answer will be option A. Next, golden rice is yellow in color due to the presence of, we know the answer will be option B, beta carotene. Next, which of the following statement is not correct regarding the genetic modification of crops? A. It makes crops more tolerant to abiotic stresses. B. It results in decreased efficiency of mineral usage by plants. C. It helps to reduce post-harvest losses. D. It enhances the nutritional value of food. So here the answer will be option B. Next, Bt-toxin genes have been expressed in plant in order to provide resistance against lepidopterans and fungi, animals and bacteria, bacteria and fungi, coleopterans and dipterans, and 5 hoche lepidopterans. So here the answer will be option D, 4 and 5, coleopterans and dipterans and lepidopterans. Next, the process of RNA interference has been used to make tobacco plant resistance to A. Bacillus thuringiensis, B. Melodygyne incognita, C. Flies and mosquitoes, D. Both A and B. Here the answer will be option B. Melodygyne incognita infects the root of tobacco plants and causes great reduction in yield. Fire and Melo in 1998 invented a novel strategy to prevent this infestation. It was based on the process of RNA interference. Next, what causes the inactive form of Bt-toxin, that is the prototoxin, to get converted to its active form in the body of an insect? A. Temperature of the gut, B. Enzyme present in the saliva, C. Alkaline pH of the gut, D. There is no specific reason. The answer will be option C. Next, Bt corn has been made resistant from corn borer disease by introduction of the gene option A. Cry1AB, B. Cry2AB, C. Ampicillin resistant gene, and D. Tryptophan. So here the answer will be option A. Cry1AB has been introduced in the Bt corn to protect it from corn borer. Next, Bt-toxins kills insect by inhibiting protein synthesis, B. Generating excessive heat, C. Creating pores in the midgut epithelial cells leading to cell swelling and lysis, and D. Obstructing a biosynthetic pathway. Here the answer will be option C. Next, Bt-toxins are intracellular lipids, intracellular crystalline proteins, extracellular crystalline proteins, and intracellular polysaccharides. What will be the answer? The answer will be option B, intracellular crystalline proteins. Next, which of the following is not used as biofertilizer? A. Bacillus thuringiensis, B. Anabina, C. Nostoc, D. Rhizobium. The answer will be option A. Bacillus thuringiensis is not used as biofertilizer, but it is used to create transgenic plants. Next, read the given statements and select the correct option. Statement 1. Food derived from transgenic crops are called as GM foods. Statement 2. Health and food safety concerns have been raised to ensure the safety of GM foods. Option A. Both statement 1 and 2 are correct, and statement 2 is the correct explanation of statement 1. B. Both statement 1 and 2 are correct, but statement 2 is not correct explanation of statement 1. C. Statement 1 is correct and statement 2 is incorrect. D. Both statement 1 and 2 are incorrect. So here the answer will be option B. Both statement 1 and 2 are correct, but statement 2 is not the correct explanation of statement 1. Next, NEET gene for nitrogen fixation in cereal crops like wheat, jowar, etc. is introduced by cloning A. Rhizobium meliloti, B. Bacillus thuringiensis, C. Rhizopus stolonifer, D. Agrobacterium tumefaciens. So here the answer will be option A, Rhizobium meliloti. Next, the transgenic plant flavor saver tomato carries an artificial gene for A. Delayed ripening process, B. Longer shelf life, C. Enhanced flavor, and D. All of these. So the answer will be option D, all of these. RNA interference involves A. Synthesis of cDNA and RNA using reverse transcriptase, B. Silencing of specific mRNA due to complementary RNA, C. Interference of RNA in synthesis of DNA, D. Synthesis of mRNA from DNA. So the answer will be option B. Next, select the correct statement regarding an improved variety of transgenic basmati rice, that is golden rice. A. It does not require the use of chemical fertilizers. B. It is completely resistant to all insect pests and diseases. C. It gives high yield but no characteristic aroma. D. It gives high yield and rich in vitamin A. So the answer is option D. Next, which of the following agricultural challenges cannot be solved with transgenic techniques? A. Crops are damaged by frost or drought. B. Crops are damaged by insect pests. C. Public concern about safety of synthetic pesticides. D. Public preference for organic vegetables. And the answer is option D. Next, read the given statements and select the correct option. Statement 1. GMO tomato flavor saver has increased shelf life and better nutrient quality. Statement 2. This is achieved by reducing the amount of cell wall degrading enzyme polygalacturonase responsible for fruit softening. So what will be the answer? The answer will be option A. Both statement 1 and 2 are correct, and statement 2 is the correct explanation of statement 1. We have discussed it previously. Next, hirudin is a protein produced by Hordeum vulgare which is rich in lysine. B. A toxic molecule isolated from Gossypium hirsutum which reduces human fertility. C. A protein produced from transgenic Brassica napus which prevents blood clotting. D. An antibiotic produced by a genetically engineered bacterium Escherichia coli. So the answer is option C. Hirudin is a protein that stops blood clotting. The gene encoding hirudin was chemically synthesized. This gene was then transferred into Brassica napus, where hirudin accumulates in seeds. The hirudin is purified and used as medicine. Next, main objective of production of herbicide-resistant GM crops is to A. Encourage eco-friendly herbicides. B. Reduce herbicide accumulation in food articles for health safety. C. Eliminate weeds from the field without the use of manual labor. D. Eliminate weeds from the field without the use of herbicides. So the answer will be option B. Weeds such as triga decreases crop yields and quality by competing with crop plants of light, water and nutrients. Weeds are to be removed with the help of herbicides or weed killers. Herbicide tolerance has been developed in maize, cotton, soybean, tobacco, etc. The main aim is to reduce herbicide accumulation in food products for health safety. Next, a transgenic food crop which may help in solving the problem of night blindness is developing countries is A. Bt cotton, B. golden rice, C. flower saver, D. Bt corn. And the answer is option B. Next, which of the following is the nematode that attacks the roots of tobacco plant? A. Agrobacterium tumefaciens, B. Rhizobium leguminosarum, C. Melodygyne incognita, and D. Tania solium. And the answer is option C. Melodygyne incognita. Next, Cry2AB and Cry1AB produce toxins that control A. Cotton bollworms and corn borer respectively, B. Corn borer and cotton bollworms respectively, C. Tobacco budworms and nematode respectively, D. Nematodes and tobacco budworms respectively. And the answer is option A. Next, first genetically modified plant commercially released in India is A. golden rice, B. flower saver, C. Bt-brijal, D. Bt cotton. And the answer is option D. Bt cotton farming has shown good results in Malwa region in Punjab. Some of the characteristics of Bt cotton are A. long fiber and resistance to aphids, B. medium yield long fiber and resistance to beetle pest, C. high yield and production of toxic proteins crystals which kill dipteran pest, D. high yield and resistance to bollworms. And the answer is option D. Transgenic Bt cotton, which produces its own insecticide, contains a gene from a soil bacterium, Bacillus thuringiensis. Bt cotton is usually resistant to bollworms disease of cotton. It is the first genetically modified crop of India. This has been developed by MAHYCO, Maharashtra Hybrid Seed Company, in collaboration with American company Monsanto. Next, 40. Bacillus thuringiensis form protein crystal which contain insecticidal protein. This protein bind with epithelial cells of midgut of insect pest, ultimately killing it. B. is activated by acidic pH and the gut of the insect or pest. C. does not kill the carrier bacterium which itself kill, resistant to toxin, and D. all of these. The answer will be option A. Next, study the following statement regarding organic farming and select the correct ones. 1. It utilizes genetically modified crops like Bt cotton. 2. It uses only naturally produced inputs like compost and biofertilizer. 3. It does not use pesticides and urea. 4. It produces vegetable rich in vitamins and minerals. Here the option will be option C, both 2 and 3. Next, the Bt-toxin is not toxic to human beings because A. the pro-Bt-toxin activation requires temperature above human body temperature. B. the Bt-toxin recognizes only insect-specific targets. C. the pro-Bt-toxin activation requires pH lower than that present in human stomach. And D. conversion of pro-Bt-toxin to Bt-toxin takes place in highly alkaline conditions. The answer is option D. Next question. Following figures show the different steps involved in production of genetically engineered insulin. Identify the enzymes involved in steps A, B, and C and select the correct statements regarding this. So study the picture carefully. And here the answer is option C. Enzymes involved in step C are DNA ligases. Next, DNA fingerprinting refers to a molecular analysis of profiles of DNA samples. B. Analysis of DNA samples using imprinting devices. C. Techniques used for chemical analysis of different specimens of DNA. D. Techniques used for identification of fingerprints of individuals. And here the answer is option A. DNA fingerprinting or DNA typing or DNA profiling is a technique of determining nucleotide sequence of certain areas of DNA which are unique to each individual. Important for DNA fingerprinting are short nucleotide repeats that vary in number from person to person but are inherited. These are the variable number of tandem repeats or VNTRs. The VNTRs of two persons may be of the same length and sequence at certain sites but vary at others. Next, what is the permanent cure of adenosine deaminase or ADA deficiency in children? Option A, bone marrow transplantation. B, enzyme replacement therapy in which functional ADA is given to patient by injection. C, infusion of genetically engineered lymphocytes in which functional ADA cDNA is introduced into the patient's blood. D, introduction of gene isolated from the bone marrow cells which produce ADA into the cells of the patient at early embryonic stages. Here the answer is option D. Next, which of the following statement is not correct? Most Bt-toxins are insect group specific. B, RNAi takes place in all eukaryotic as well as prokaryotic organisms as a method of cellular defense. Next, C, the recombinant therapeutics do not induce unwanted immunological responses in humans. D, adult onset diabetes can be controlled by taking insulin at regular time intervals. And the answer is option B. Next, given figure represents the maturation of proinsulin into insulin. Identify the product A. A, polypeptide chain A, polypeptide chain B, polypeptide chain C. Option D, both B and C. And the answer is option C, polypeptide chain C. Next, early detection of a disease is possible by option A, PCR, B, gene therapy, C, recombinant DNA technology and ELISA, and option D, both A and C. And the answer is option D, using conventional methods of diagnosis, serum, urine analysis, etc., early detection is not possible. Recombinant DNA technology, polymerase chain reaction or PCR, and enzyme-linked immunosorbent assay or ELISA are some of the techniques that serve the purpose of early diagnosis. Next, adenosine deaminase deficiency can permanently cured by which of the following methods? Bone marrow transplantation, enzyme replacement therapy, gene therapy at early embryonic stage, and option D, all of this. And the answer is option C. Next, technique used to detect the DNA in a clone is option A, PCR, B, gel electrophoresis, C, chromatography, D, autoradiography. And the answer is option D. A single-stranded DNA or RNA joined with a radioactive molecule or probe is allowed to hybridize to its complementary DNA in a clone of cells. It is followed by detection using autoradiography. The clone having the mutated gene will not appear on photographic film because the probe will not have the complementarity with the mutated gene. Next, it is a single-stranded DNA or RNA tagged with a radioactive molecule and is used to detect mutated genes. A, RNAi, B, probe, C, plasmid, D, primer. And the answer is option B, probe. Next, which of the following statement is not correct? A, insulin used for diabetic patients was earlier extracted from pancreas of slaughtered cattle and pigs, which was more efficient than the genetically engineered insulin. B, PCR technique is applied to detect HIV in suspected AIDS patient and to detect mutations in genes in suspected cancer patients. C, bone marrow transplantation requires periodic infusion of genetically engineered lymphocytes in ADA deficient patients. D, bioremediation is one of the applications of biotechnology. And the answer is option A. Earlier, insulin used for carrying diabetes used to be extracted from pancreas of slaughtered pigs and cattle. This insulin was slightly different from human insulin and caused some undesirable side effects such as allergy. Genetically engineered insulin is far more efficient. Next, what might be an advantage of beginning gene therapy prior to birth? A. This would give the body plenty of time to utilize the new genes. B. The body would not reject it as it has not yet recognized self. C. The cell being extremely young are more receptive of gene therapy. D. There probably is not any advantage. And the answer is option B. Next, which of the following companies started selling humulin in the year 1983? A. Eli Lilly, B. Gentech, C. GAC, D. None of these. And the answer is option A. In 1983, Eli Lilly, an American company, first prepared two DNA sequences corresponding to A and B chains of human insulin and introduced them in plasmids of Escherichia coli to produce insulin chains. Chains A and B were produced separately, extracted, and combined by creating disulfide bonds to form human insulin or humulin. Next, which of the following statement is incorrect about gene therapy in ADA deficiency? A. Lymphocytes from patients' blood are taken out and cultured. B. A functional ADA cDNA is introduced into these lymphocytes. C. Lymphocytes are then introduced in the body of patient. D. Patient does not require periodic infusion of genetically engineered lymphocytes. And the answer is option D. It is the incorrect answer. As a first step towards gene therapy, lymphocytes from the blood of the patient are grown in a culture outside the body. A functional ADA cDNA using a retroviral vector is then introduced into these lymphocytes, which are subsequently returned to the body of the patient. However, as these cells are not immortal, the patient requires periodic infusion of such genetically engineered lymphocytes. Next, the study of all proteins coded by the genome is called as proteomics, genomics, gene library, proteology. And the answer is option A, proteomics. Next, gene therapy can be referred to as A. Preclinical testing for inherited diseases in newborns. B. Treatment of diseases caused by genetic defect. C. Genetic engineering using rDNA technology. D. Cancer treatment using in vitro cultured stem cells. And the answer is option B. Next, figure given below depict the procedure for gene therapy. Pick up the disorders for which this technique has been applied successfully. Study this picture carefully. Option A, adenosine deaminase deficiency. B, AIDS. C, myasthenia gravis. D, both A and C. And the answer is option A, ADA deficiency. Next, an example of gene therapy is A, production of injectable hepatitis B vaccine. B, production of vaccines in food crops like potatoes which can be eaten. C, introduction of gene of adenosine deaminase in persons suffering from SCID. D, production of test tube babies by artificial insemination and implantation of fertilized eggs. And the answer will be option C, introduction of gene for adenosine deaminase in persons suffering from SCID. Next, human insulin is being commercially produced from a transgenic species. A, Mycobacterium. B, Rhizobium. C, Saccharomyces. D, Escherichia. Answer is option D. Some of the steps involved in production of humulin are given below. Arrange them in correct sequence and select the correct option. Here the correct sequence is option C. Next, during the processing of proinsulin into the mature insulin, option A, C peptide is added to proinsulin, C peptide is removed from proinsulin, B peptide is added to proinsulin, B peptide is removed from proinsulin? And the answer is option B. Next, study the following steps which are followed during the process of gene therapy while treating a patient of SCID. 1. Retrovirus infects lymphocytes extracted from bone marrow of the patient and cultured. 2. Engineered cells are injected into patient's bone marrow. 3. Normal allele is inserted into a retrovirus. 4. Retrovirus makes a DNA copy of its RNA. This DNA carrying the normal allele gets inserted into the chromosome of the host cell. Arrange the above given steps in correct sequence and select the correct answer. And the answer is option B. For effective treatment of a disease, option A, early diagnosis is required but understanding of its pathophysiology is not required. B, early diagnosis not required but understanding of its pathophysiology is required. C, early diagnosis and understanding of its pathophysiology are required. D, neither are the early diagnosis nor understanding of its pathophysiology is required. The answer is option C. For effective treatment of a disease, early diagnosis and understanding its pathophysiology is very important. Using conventional methods of diagnosis, early detection is not possible. Recombinant DNA technology, PCR, ELISA are some of the important techniques that serve the purpose of early diagnosis. Next, a doctor while operating on an HIV positive patient accidentally cuts himself with a scalpel, suspecting himself to have contracted the virus. Which test will he take to rule out or confirm his suspicion? Option A, PCR. B, routine urine examination. C, TLC. D, DLC. And the answer is A, PCR. Very low count of bacteria or viruses when the symptoms of the disease are not yet visible can be detected by multiplication of their nucleic acid by PCR. PCR is usually used to detect HIV in suspected AIDS patient. PCR can detect very low amounts of DNA. Next, second generation vaccines are prepared by recombinant DNA technology. Which out of the following are the examples of such vaccines? A, hepatitis B virus vaccine. B, herpes virus vaccine. C, SOX polio vaccine. D, both A and B. And the answer is option D. Second generation vaccines are vaccines which are prepared by recombinant DNA technique or genetic engineering. Example, herpes B virus vaccine, herpes virus vaccine and pneumonia vaccine. The yeast-derived recombinant hepatitis B virus vaccine is the first commercially available human vaccine produced by the genetic engineering technology. Hepatitis B vaccine is produced from transgenic yeast by recombinant DNA technology. Animals that have had their DNA manipulated to possess and express a foreign gene are called transgenic animals, somatic hybrids, soma clones, super animals. And the answer is option A. Next, a human protein which is being obtained from transgenic animals and is used to treat emphysema is option A, alpha-lactalbumin, B, thyroxine, C, alpha-1 antitrypsin, D, insulin. And the answer is option C. Next, which of the following is not benefit of transgenic animals? A, investigation of new treatment for diseases. B, early detection of diseases. C, testing the safety of vaccines. D, to produce useful biological products. And the answer is option B. Next, match the column 1 and column 2 and select the correct answer from the codes given below. Here the answer will be option B. Next, Dolly sheep was genetically similar to A, the mother from which a nucleated egg cell was taken, B, the mother from which nucleated udder cell was taken. C. The surrogate mother. D. Both surrogate mother and donor mother. And the answer is option B. Dolly the sheep was cloned in 1997. Ian Wilmot and his colleagues at the Roslin Research Institute in Scotland took cells from ewe mother sheep's udder. They stored these udder cells in low nutrient culture. This stopped the starved cells from dividing, that is in the G0 phase, and switched off their active genes. One complete udder cell with its nucleus was selected because its nucleus carries the mother genetic information. In the meantime, unfertilized egg cell was taken from a different ewe. Its nucleus was removed, leaving an empty cell containing all the essential machinery to produce an embryo. They then fused udder cell nucleus with the empty egg cell by electrical stimulation. The altered egg was cultured for 6 days. Out of many resulting embryos, one was implanted in the uterus of the surrogate mother, where it grew into a lamb. So Dolly was born, genetically identical to mother sheep because her first cell nucleus came from mother cell. Dolly's birth proved that adult cells could acquire the cloning potential of embryonic cells. Next, the transgenic animals are those which have A. Foreign RNA in all its cells, B. Foreign DNA in some of its cells, C. Foreign DNA in all its cells, D. Both A and C. And the answer is option C. Transgenic organism is one that has become transformed following the introduction of novel genes into its genome. It is most frequently achieved by integration of cloned DNA sequences following their injection into the fertilized egg. This fertilized egg divides mitotically to form the whole organism so that all the cells of the organism will carry the transferred gene. The transferred genes are known as transgenes. Next, match the column 1 containing transgenic organism with their specific characteristics in column 2 and select the correct answer from the codes given below. And here the answer will be option A. Next, read the given statements and select the correct option. Statement 1, transgenic mouse is termed as super mouse because it is twice big in size than normal mouse. Statement 2, in super mouse the gene for human growth factor has been introduced and expressed. Here the answer will be option A. Both statement 1 and 2 are correct and statement 2 is the correct explanation of statement 1. Next, genetic engineering has been successfully used for producing A. Transgenic mice for testing safety of polio vaccine before use in humans, B. Transgenic cow-Rosie which produces high fat milk for making ghee, C. Animals like bulls for farm work as they have superpower, D. All of this. And here the answer is option A. Transgenic mice are being formed for use in testing the safety of vaccine before they are used on human beings. They are being used to test the safety of the polio vaccine. The first transgenic cow, Rosie, produced human protein enriched milk. The milk contained the human alpha-lactalbumin. It is a more balanced product for human babies than natural cow milk. Bulls with more power are produced by animal breeding, not by genetic engineering. The organization which makes decisions regarding the validity of GM research and the safety of introducing GM organisms for public service is A. Genetic Engineering Approval Committee, B. Genome Environment Action Committee, C. Genetic Environment Approval Committee, D. Genetics and Ethical Issue Action Committee. And the answer is option A. Next, A monopoly granted to a person who has either invented a new and useful article, made improvement in an existing article, or invented a new process of making an article is called a biopiracy, B. Bioethics, C. Patent, D. Genetic modification. And the answer is option C. A patent is the right granted by government to an inventor to prevent others from commercial use of his invention. When patents are granted for biological entities and for products derived from them, these patents are called biopatents. Primarily, industrialized countries like USA, Japan, and members of European Union are awarding biopatents. Next, use of biosources by multinational companies and other organizations without proper authorization from the countries and people concerned without compensatory payment is termed as A. Research partitioning, B. Biopiracy, C. Patenting, D. Biofortification. And the answer is option C. And the option is the right answer is option B, biopiracy. Biopiracy is exploitation of bioresources of a country by organization and multinational for commercial purpose with or without patent but without any access and benefit sharing agreement ABA. Bioresources or biological resources are all those organisms which can provide commercial benefits. They are abundant in developing countries which are poor in technology, though the countries have traditional knowledge related in bioresources. On the other hand, developed countries are poor in bioresources but are rich in technology. Traditional knowledge helps in saving time, effort, and expenditure in developing refined product for commercialization. Therefore, based on traditional knowledge, institutions and companies of industrialized nations are collecting and exploiting bioresources of other nations by getting them patented. Next, match column 1 with column 2 and select the correct answer from the quotes given below. Here the answer will be option C. Next, rules of conduct that may be used to regulate our activities in relation to the biological world is called bioethics, biowar, biopatent, and D. Biopiracy. Here the answer will be option A, bioethics. Next, which Indian plants have either been patented or attempts have been made to patent them by Western nations for their commercial use? A. Basmati rice, B. Turmeric, C. Neem, D. All of these have been targeted. Here the answer will be option D. India is a country rich in tradition and expertise in natural medicines, spices, food preparations, biological pesticides, and diverse agriculture. Through patenting without consent, foreign companies have collared at least 22 plants for their beneficial derivatives. Patents have been taken out on plants such as black pepper, basmati rice, Indian mustard, pomegranate, turmeric, and neem. US, Japanese, and German companies are the principal patenting pirates. Biopiracy means a use of biopatents, B. Thefts of plants and animals, C. Stealing of bioresources, D. Exploitation of bioresources without authentic permission. And the answer is option D. Next, biopatents are 1. Right to use invention, 2. Right to use biological entities, 3. Right to use products, 4. Right to use process. And here the answer will be option D. Next, X is the right granted by government to an inventor to prevent others from commercial use of his invention, where X are granted for biological entities and for products derived from them. These are called Y. Read the above paragraph and identify X and Y. And here the answer will be option A, X patent, Y biopatent. Next, which of the following has been covered under the broad patent category? A. Triticum, B. Oryza, C. Pisum sativum, D. Brassica. And the answer is option B, Oryza. Which of the following statement is correct? A. The current interest in manipulation of microbes, plants and animals has raised serious ethical issues. B. One possible risk of genetic engineering is the accidental production of antibiotic-resistant microorganisms. C. Although risks are possible, genetic engineering offers more of contribution to human welfare than threats. D. All of this. And the answer is option D, all of this. Which variety of rice was patented by a US company even though the highest number of varieties of this rice are found in India? A. Sharbati Sonora, B. CO 667, C. Basmati, D. Larma Rojo. And the correct answer is option C. In 1997, an American company got patent rights on basmati rice through the US Patent and Trademark Office. This allowed the company to sell a new variety of basmati in the US and abroad. This new variety of basmati had actually been derived from Indian farmers' varieties. Indian basmati was crossed with semi-dwarf varieties and claimed as an invention or a novelty. The patent extends to functional equivalence, implying that other people selling basmati rice could be restricted by the patent. Next. Which step has been taken by Government of India to cater to the requirement of patent terms and other emergency provisions in this regard? A. Biopiracy Act, B. Indian Patents Bill, C. ETI Act, D. Negotiable Instrument Act. And the answer will be option B. The Indian Parliament has cleared the second amendment of the Indian Patent Bill. It takes into consideration unauthorized exploitation of bioresources. It also involves patent terms, emergency provisions, and research and development initiative. And the last question. Which of the following statements is correct regarding Genetic Engineering Approval Committee, GEAC? Option A, it makes decision regarding the validity of GM research. B, it ensures the safety of introducing GM organisms for public services. C, genetic modification of organisms can have unpredictable results when such organisms are introduced into the ecosystem. Therefore, the Indian government has set up organizations such as GEAC, and option D, all of these. And here the answer will be option D, all of these. Now, time for some extra questions. But I will not discuss these question answers. You have to try it yourselves. You guys understand it very well. The video is become very long. Sorry for this long video, and thanks for watching.